that the number 5 slot, so Steve Ellsby from RCUK is going to be added. That means we have 6 speakers in an hour. So forgive me, but I'm going to need to insist that we move along and that we keep questions relatively short. But I would like to introduce first Rob Miller from the NSF Bio.
Okay. All right. I'll use this one. Okay, thanks. So I'm here to represent NSF, but I'll also point out that there are at least 2 other colleagues of mine from NSF in the audience that I know are here today, and that's Dr. Bill Zamer in the back. Bill is the acting Deputy Division Director for Integrative Organismal Systems, and you all already know that Steve Ellis is here, a Program Director in iOS, because he was speaking earlier on. Or commenting earlier on.
You want your slides?
That's okay.
Okay.
Okay. So I'll just give you a bit of an overview of NSF to remind you what NSF is, and we'll talk about some specific programs. And so NSF is an independent federal agency, which basically means we're not associated with any of the federal departments like agriculture or health and human services. And NSF was set up in 1950 to basically support all fields of fundamental science and engineering. NSF, for those of you who aren't familiar, is divided up into directorates. Within the Directorate for Biological Sciences, where there are 4 divisions. 3 of the divisions are focused on areas, specifically on areas of research by scale. So you have the Division of Molecular and Cellular Biosciences at the sort of smallest scale, cellular and molecular, and then the Division of Environmental Biology at the ecosystem level scale, the largest of the scales. And then in between is the division that I'm associated with, along with Bill and Steve, which is Division of Integrative Organismal Systems, sort of whole organisms and their interactions and biology. There's also the 4th division, which is the Division of Biological Infrastructure. And what I should have added after hearing some of the discussions this morning is some of the contributions that DBI makes that are very relevant probably to this community that I didn't have in the slides initially. And it's, for example, it's within DBI is the ABI program, which is Advances in Biological Informatics. So this is a program within DBI, or the ABI program, that's, its stated goals, development of tools and resources in the area of informatics. So this is something that's probably important for this community to know about if you don't already. Also, what's been really exciting for me as relatively new to the division to be learning more about how the iPlant program, which is also funded out of DBI, is now getting utilized by some of the animal community, including people sitting in this room. And I've been hearing about that this morning. So I think talking to some of the both investigators and some of the program directors that are here that are familiar with the iPlant program and the resources that provides for to the community is something that you should be learning more about. It's set up for the plant genome community, kind of a throwback to what we were talking about this morning. The Sunny mentioned that the sort of how the plant genomics community was sort of a step ahead. That was one of the tools that was developed as part of that, but the infrastructure, the platform there is amenable to other broader areas outside of plant genomics. So within the Directorate for Biological Sciences, there are a number of initiatives on the way that are relevant to this community as well. Coming from the Office of the Assistant Director at a directorate-wide level is genomes to phenomes, which has been is a theme running through this meeting that we've talked about, and also a relatively newly developed concept that's going to be rolled out over the near future called URL, or Understanding the Rules of Life. This is something that Jim Olds, who's our Assistant Director for Bio, is wanting to take as an initiative that is across the Directorate in Biology, which is getting at the basic principles of life on this planet, the rules of life. There's also division level initiatives that hopefully will reach out to other divisions and even other directorates in NSF. That's one of the fun things that can happen in NSF. But one that's about to be announced, I would say within the next, hopefully, couple of weeks, there will be information on the street about it, is a program that we've been formally talking about in the division for at least a year, but I can remember when I was a program director 8 years ago, us having concepts along this line, which is a program called EDGE, which is enabling discovery through genomic tools. And this is going to cross the core programs, certainly within our division, and hopefully reach out to other programs and other divisions within NSF. That is addressing a concern that we're facing with a lot of the PIs that the proposals that come in, that they're getting to the point where they've got their transcriptomes, they've got their phenotypes, but it's hard to make the causation connections between those two data sets, which is really a lot of what's been talked about at this meeting as well. Enabling model organisms in ways that they haven't been enabled before, or providing the tools. And these are going to be proposals that we're hoping will be getting in the fiscal year or calendar year 16 for funding in fiscal year 17. So this is a new program to come along that I think this community would find that they of interest to them. So then we got sent 3 questions to answer. And so, is understanding and predicting complex phenotypes, genotypes, and epigenetic data a priority for NSF? Yes. This is part of our Genomes to Phenomes program. There's going to be a Genomes to Phenomes workshop being funded, or it's already in the works. In fact, it's later this month. This is co-funded from 2 bio divisions, ours, iOS, and MCB, along with folks in the Division of Mathematical Sciences and in MPS, another directorate. So it's aimed at multi-scale modeling to get at complex biological systems. And also what I failed to put on here is that within our division was funded a research coordination network called the Genomes to Phenomes Network by Carl Schmidt, who I think I've seen is here at the University of Delaware, and Steve Ellis was the managing program director for that research coordination network, and so a funding of a program through our RCN mechanism. Second question, could the functional annotation of domesticated animal genomes fit into that priority? NSF's priority does not preclude the use of domesticated animals. It depends on the nature of the question. And I think I should wrap this up quickly because I'm going to be running out of time here. But, EDGE activities like we just talked about, there's nothing that precludes them from being domesticated animals. It's the nature of the questions getting asked, whether or not they're getting at fundamental biological principles. And 3, what are possible mechanisms? So you heard earlier today, for funding these sorts of things, you heard earlier today about a joint solicitation that will be going out between the USDA, NIFA, and NSF iOS against plant-biotic interactions. So there's opportunities for developing potential interagency programs such as that. There's also examples of, again, this is where the plant folks are kind of ahead of the curve, developing programs with private foundations. There's a program called BREAD that is a joint NSF and the Gates Foundation program that also facilitates a lot of international funding, international collaborative agreements, the sorts of mechanisms that a lot of the agencies use, and a program that's been very useful called the Ideas Labs, which brings together people to really create initiatives in particular areas. So there's the three of us are around here all day, and hopefully there'll be a chance to talk, but I've probably used up my time after being told to keep it short.
Okay, so we've got 2 minutes literally for questions here, so I'm going to try to move quickly.
Just if I remember correctly, NEFA also has invested in iPlant. Is that correct, or am I misunderstanding that?
But we funded a couple of grants towards the soil. Use of it. So the projects that got funded essentially would leverage the iPlant infrastructure in order to do more animal-oriented things, but it's not directly to iPlant itself.
With respect to joint solicitations, could you maybe talk a little bit about some of the challenges and opportunities in developing a joint solicitation and kind of what it takes to get there? Because that seems to be a really good model of leveraging dollars.
Right. So I think one of the important things that in terms of developing it was recognizing in both communities, both funding and both agencies at the program director level, that there was a significant overlap in the PI community, that we were using the same panelists, we were using the same reviewers, I used to be a program director in the program that this one's emerging from, which is at NSF is SDS, Symbiosis, Defense, and Self-Recognition, and then in the plant disease side of NIFA. And so it became obvious that there was this community, and you can see it sometimes too. You've got, investigators out there. This is my NSF project. This is my USDA project, and I frame my proposals in that perspective. So recognizing that maybe the opportunity then exists within these communities, that in fact you could even encourage proposals that go from one end to the other in the same proposal. And there's ways, that at the NSF side, certainly, and I think that must exist at the USDA side as well, there's a lot of discretionary power at the level of the program directors to say, well, we'll fund this part of this proposal, and you can fund that part of the proposal, right? And those mechanisms exist. And that allows the PIs then to write something that maybe is a bit more cohesive too. So that's not going to preclude, there's going to be proposals coming in that are NSF only, really, or really just US or NIFA only, should be funding the whole thing. But we're hoping to see that continuum between them. So it takes the MOUs between the agencies, those are being set up, and it takes clearing solicitations. I mean, the nuts and bolts of it, clearing solicitations between the upper offices of both agencies as well too. And that's, but it, it starts with that recognition of what's going on in the community too. And I think that really worked out well in this case.
Thank
you. Yeah.
Thank you. I'm sorry, I'm going to have to move on quickly. We may have some time at the end. If we do, we can readdress some more questions. Please join me in thanking Dr. Miller. I'd like to present Dr. David Bailey from Canadian Genome Enterprise.
Well, first of all, thank you, Chris, for inviting me. And so I'm going to try to answer those three questions in some examples of what we are actually already funding in annotation area, and also talk about an opportunity really to address that last question about funding and opportunities within Canada for multinational kind of collaborative studies. So I want to get to that and hopefully have some time for questions after. So I'm going to, I know not everybody's familiar with the Genome Canada model, and I'm really representing Genome Alberta. That's my home. I'm the CEO for Genome Alberta, but we have a partnership agreement across Canada with the 6 genome centers, and Genome Canada, our federal partner. So it's a little hard to read. I know it's small, but there's 6 of us, and so we have different areas of strength. Alberta's obviously livestock and crops are a big area of our interest, and particularly my interest. And we maybe not as strong in human health, but I want to mention these are the areas. So unlike a lot of you in the room where you're very focused on the animal area, our areas of funding go across all of these sectors that are important to us in some and varying degrees. aquaculture and fish area is not really that as important in the inland area like where I am, but mining and energy and environment are huge topics for us, in addition to the agri-food area and, of course, human health. As far as grants and projects, there's two really main ones that Genome Canada supports. One is the large-scale competition, which is really open and what it requires a Canadian anchor. And the second is really an industry-driven competition. It's called GAP, Genomics Applied Partnerships Program. And we really moved towards that in a big way, a much more rapid turnaround time. But the key requirement there is the industry partner needs to be the lead on it, and not the academic. The academic is part of the support team. These strategies have all been developed through levels of communication across the country, and brought in lots of people, and they're available on the website. You can see these documents there, and they're kind of the guiding principles, and they are currently being updated, but they're still fairly current in terms of large-scale competitions. So the large-scale competitions, this is a little bit of a history of it. In 2012, it was a genomics and personalized human health one. 2014 was really an agri-food focus, feeding the future. And we currently, in the midst of natural resources and the environment, so natural resources, forestry, energy, mining, and the environment. And to a lesser extent, there's an opportunity for, I'd say, species that are, I'd say, are not really looked at very closely. So often the ones that are more rare species, there's an opportunity for that. And prions. Prion was thrown in this because of the work that we've done on prion research, particularly starting with BSC, but then with chronic wasting. So those are the eligible areas that are that's current in play right now. The next campaign are ones depending on funding. So the intent is hopefully to have new funding for a human health competition in 2016, and then we will repeat that cycle, agriculture and then natural resources. So the large scale, these are the goals, and I know the slides will be available, so I'm just going to slip through these quickly. This is the large scale ones, where two were funded in 2010, one in beef and the other in swine. What's important here is the example, and to get to the topic of funding opportunities in the future. These are multi-institutional, multi-country projects in many cases. So I said it needs a Canadian anchor because of the public investment in it, public dollars. But we're looking for the best possible teams to work together. So the second project was this one, originally led by Steve Moore, and then when he moved, Steve Miller, and then when he moved, Paul Stoddard. But anyway, an example of the collaborations. And so these are the overall objectives. This is a list of the projects that were funded. So I showed you the two that I actually managed through Genome Alberta, but across Canada, here are the ones that were funded. So when we say large scale, that's roughly 10 million, and why that number is it's rough. I mean, there's variation there, but the reason is that in the Canadian granting system, there's a number of different granting opportunities through different agencies. But Genome Canada needed to separate themselves from those. And so the other granting councils are usually much, much lower dollars per project. They're in the $200,000 to $300,000 projects, whereas Genome Canada, differentiated itself by saying these are expensive, big projects, international players, and this is the level of funding roughly we're looking at. Now the new model is really, it's always a co-funding model. So whatever you see on there, the Genome Center has to find half of those funds, or collectively with the principal investigators come up with half of those dollars. And in-kind does count, but to a certain level. So here are the two new projects that were just recently funded this last summer, and kind of an example of the leads and who to contact on that. So this happens to be an Ontario-based project, but they chose to run it through Genome Alberta, and luckily it was successful. So we have that level of, I'd say, flexibility. So the other thing, so Listeria is one project. So this was a national initiative, want to move to, sometimes these projects are much more grassroots and initiatives start at the regional level. So this is an example of one that's funded, and you see some of the players on there. Did not have Genome Canada funding in it, but we're able to raise the funds regardless. So the Genome Canada model, we are not simply tied to that. E. coli, so again, a regional initiative and a food safety topic that's really important. And this one's generated some really, so 18 months, so the turnaround time is much more quickly, because it was, the desire here is how do we utilize these tools and put them out there. And so it was good. So funding is not just, as I said, we managed it, but one went to Laval and the other one is in Alberta, the two projects here, and started with scratch, didn't have $1 for that, said it was an important topic, and we raised almost $2 million for that. So, and this is the last one, and I'll quit then, and just PED, again, was a great project to get involved with. Worked with the National Pork Board to make sure that the projects were complementary. Their time frame was 6 months and ours was 18 months. So different types of project, and it worked really well. So I think I should mention that the competition that Genome Canada will, and you go to their website for this, but it's on bioinformatics and computational biology, and look forward to international collaborations on that and putting forward those proposals and getting external reviewers like Jerry into the projects as well.
Thank you very much. Are there questions? Okay.
I guess I could ask a question. So, David, if I could characterize the projects, that they have at least there's a fairly significant applied focus. Would Genome Canada consider something where it might be developmental, an informatics project, or some project that doesn't have a foreseeable product at the end or improvement of product at the end? Most of the project I'm familiar with, it's fairly well defined that you're improving the animal or improving the trait or whatever.
Yeah, so I think the examples we've used so far are about the applied ones, are really an end user, the pool is there, but the bioinformatics and computational biology competition will be exactly what you're talking about here, much more fundamental work that needs to be done, and how do we make sure that there's a cross-talk between the different institutions on data collection and retrieval and storage and all of that. Great. Okay,
thank you. And next up is Dr. Parag Chidnis from USDA NIFA.
Okay. Good morning, and I'm here representing NIFA, which is one of the agencies at USDA, and we have other sister agency, Agricultural Research Service, and many of the participants here are from that agency too. So if you have specific questions about how ARS functions in the animal genomics area, they'll be here, and maybe after my talk, if you have questions about that, you can ask those too. But my talk is particularly on NIFA, which is the extramural funding agency. So it's about $1.5 billion budget, and it gives both competitive and capacity grants. What it means, about half of its budget is divided among the land grant institutions, where then the institutions decides how that money is invested in research on those in agricultural experiment stations. And then the remaining half is funded through various competitive programs, where then the peer review system decides where the funding goes. So there is a partnership with the land grant institutions in the United States, where then we work together to figure out how the agricultural research will get funded. So just to firstly, the questions that were sent to me, I think the answer to all of those is yes. So I can just then skip those questions and then move on to some of the ideas. Yeah. NEFAS funding in animal genomics currently is through either capacity funds, and those are also very important. For example, multi-state research projects we have. One of our exemplary research project is on animal genomics, meets every year at PAG, and many of the participants here are from that project. And I think Eric Young is here also, who is the administrative, I think, liaison to that project. And then we have a number of hatch projects, and this is about $9 million investment in specifically in animal genomics. And then we have a number of different computative programs. Most of the funding comes from foundational grants and our flagship program called AFRI. And in that particular one, then the grants are about $500,000, and there are several components of FANG that have been funded through that program on a smaller scale, like about 500K type of projects. Then there is a food security challenger area, which is particularly relevant to this community, where plant and animal breeding is one of the priorities. It was in 15, it is in our budget request in 16, and that's one of the area where this type of research could fit well. Then we also fund, when we talk about animals, we not only just farm animals for food production, it's also aquaculture. So we have also some aquaculture programs that where even genomics projects, genetics breeding projects get funded. If you need to know more about AFRI, I think we have many people here from NEFA, including Mark Mirando, who is Mr. AFRI and knows everything about this program. Okay. So in AFRI investments, about 20% of the budget goes to animal sciences, including both production and health. And part of that, so just to give you context, this is about a $325 million program where about 20% goes in this area. Part of that goes in animal genomics. So that's kind of a context how animal genomics fit in the big picture at NEFA. Looking at the genome to phenome and the opportunities, this is something really an important area for us at NEFA. When we look at how the information in the genomes is eventually going into the phenotypes that are important for agriculture. So when we are thinking about this kind of understanding, deciphering this information, there are, I think, a lot of great opportunities in terms of understanding the rules of life that you just heard from Rob when he was talking about NSF's priorities. You also, I think there are, a lot of opportunities for applications. For example, say accelerated and predictive breeding, whether it is in crops or in animals. And I think this is the second part, this is the application part, is where I think somebody like USDA is really very interested in how these kinds of transformative, I think, platforms or discoveries will help in applications that will impact American agriculture. Whereas the rules of life type of discoveries probably are more important, where the basic science, fundamental science is valued with, like at NSF. So I think when you are thinking about how this transformative kind of change from understanding phenotype from genotype comes, there are a number of challenges, including perhaps theoretical underpinnings of how do we really dissect that black box of going from linear information to the scales that were really well described yesterday in the plenary talk, from going to the genome, like DNA sequence, to the genome-wide kind of epigenetic information, to cellular information, organismal information. And how do we really get to that? And then when probably we'll need for a lot of technologies where you need to have computer scientists, you need to have engineers involved in order to really try to get high throughput data and decipher it. So data resources, cyber physical, I think, systems, and you have examples of how animal scientists are working with iPlan to use those systems. And then data analytics, so that there are predictive algorithms that could be used. So there are a lot of challenges intellectually, not just for this community or biologists, but also for computer scientists and engineers, which can really help them get excited about this challenge. So when we look at this genome to phenome in terms of, I think, the area and interests of different agencies represented here, it's really a ripe area for interagency collaborations, because the potential outcomes of this area are of interest to everybody. If you look at this, how the research sometimes can get divided into potential kind of like the applications versus fundamental understanding, the Bohr's kind of quadrant, what people call for basic research, where NSF is interested in, where they want to find out how phenotypes in nature are determined by genotype, what are those rules. NEFA is interested in agricultural phenotypes, how they are determined, what are the applications. NIH is interested in human phenotypes, how they are important. And there will be industry in the very applied research area, which will be interested too, including the animal breeding industry. So there are, I think, interests around in different in US agencies as well as in EU, Canada, as we just saw, where I think it's ripe for collaboration. So what I thought we could do is kind of maybe look at some of those where I'm very familiar with in terms of interagency collaborations that were developed. There are many other examples, like, and others can give you examples like cattle genome sequencing and other types of projects. Couple of those, the first two, which was Arabidopsis functional genomics and National Plant Genome Initiative, where I think several agencies are working together. They started in an era where Congress was very liberal in giving funding increases. So it was, I think, possible at that time to put large amount of money. The other two, that's synthetic genome and international VTL partnership, those are the projects that are in this fiscally kind of tough era currently, where Congress is very, in the US, is not as liberal in giving funding increases. But what actually helped in those projects, they were as an idea where I think a lot of agencies got interested because those were going to make breakthroughs, help industry and potential transformation in bioeconomy. And that's why then they got interested, and then many of the agencies, in first case, like NSF, China's Ministry of Science and Technology, BBSRC, the funding agencies from Singapore, Australia, and others have now joined to come to synthesize entire yeast genome. In the second case, I think BBSRC, CEMIT, USAID, ARS is also part of it, then NIFA, and then the Mexican funding agency, they are part of it in really trying to increase the wheat yield. And these kinds of focused projects with specific goals that a lot of agencies can look into have been like started, and it is possible to work with other agencies. And again, here we have people like Peter Johnson in the audience, who has been working with BBSRC all the time and has really good interactions with them in starting new programs. Then Mark has been working with NIH and with the dual purpose research, where also, again, we have strong collaborations. So there are different these kinds of activities, even in this fiscally conservative area times. We can that can be visualized. So these are kind of my 2 cents worth in terms of this, and I'm purposely hiding USDA logo from it because they are my personal opinions. So in these kinds of initiatives, what one really needs is like ambitious goals that are relatable to every people, right? How it's going to impact when there is someone going and, like when Sunny was here before, he's going and talking to the Ag Appropriations Committee. How can he sell that idea? So those kind of relatable goals are something that are important. Clear milestones that are measurable, because people are interested, okay, if money is invested in this project, what will we get out of it? What is also important is those economic impacts, even though currently the project is, okay, we want to find out epigenomics of cattle and all different farm animals. What are the potential breakthroughs that will come along in applications? And then the stakeholder buy-in is something that is also very important. For example, in the era, like for USDA, NEFA, it's like agricultural experiment stations. How they are buying into these projects. They have their group, like we work closely with Association of Public and Land Grant Universities. When they project, what are their priorities? How does that fit in their priorities? And again, that's something that you may want to think about. And then the different commodity boards and groups, they work with USDA all the time. How does that fit in their priority? How supportive they are of this research is important. And then the professional societies, some of the professional societies have come behind, and we say, for example, meet with American Phytopathological Society all the time, where they are trying to really promote phytobiome project, right? So how does this priority fit with the professional societies? And I think those are some of the ways you can influence what happens in Washington. So these are just some thoughts, and I'll be happy to take any questions. And if you have questions specific to ARS, which has a lot of resources in animal genomics area, I think Steve and many other people from ARS are here. So thank you.
Thank you. Okay, we're a little behind on time. Is there one quick question that someone would like to lead with? I'm sure there are many for this. So for the sake of time, let's continue those discussions afterwards. Next up is Dr. Ravi Ravindranath from NIH, who is a director of Pre-Implantation Genetics and Development Program.
Good morning. My name is Ravi. I am from the Fertility and Infertility branch at the National Institute of Child Health and Human Development. And this is one of the 24 institutes within the National Institutes of Health. So I'll start directly answering the questions. So is Fang important for the National Institutes of Health? So I'm using some historical facts here. So associating sequence variation with quantitative phenotypes has been done in domestic animals for a few decades. You all know that. So the importance of that is basically, animals are food sources and models for human health and disease, and also for ecology. So in 2002, there was a meeting at this venue for exploring horizons for domestic animal genomics. So some of you might have attended this meeting. So the perspectives given by NIH, several program officers from the National Institutes of Health participated in this meeting. And then the perspectives at that meeting were sequencing is absolutely essential for basic scientific understanding, and sequencing is essential for human health and medical research. So these were the perspectives, and these perspectives are still valid. So it hasn't changed. The reasons for that was understanding of human biology or physiology is based on domestic animal biology. So that's the most important thing. So in fact, chicken embryology was used actually to establish all the human embryology. And then several methods were developed in animals, that's IVF and cloning. That's an example. And then disease models, for example, influenza. So these were the ones that were discussed at that meeting. And like I said, it's still valid. So NIH is absolutely interested in domestic animal genomics. So what happened after that meeting was NIH did participate actually in cattle genome project and horse genome project and chicken genome project. So these were the three domestic animal models in which NIH did participate. There were other agencies which participated as well, including USDA. If you want more information, actually I have my colleague, Dr. Elise Feingold. She is here from NHGRI because this was not funded through my institute. So although there is no direct involvement with swine genome project, NIH has always been interested in swine as a model. In fact, most of the funding that goes through NIH to the domestic animal model, swine is on the top of the list. Because its growth, development, and disease pattern is similar to human. So several individual projects have been funded. So I'm not going to go to the details of those because there is enormous funding that goes into many of these domestic animal model systems right now. So what are we interested in from NIH perspective? So we are using them as models for physiology. That's the number one, top of the list. Then the second is nutrition. Third is pregnancy and fetal growth. Most of that is funded through my institute. Immunology, organ transplantation, and infectious diseases. So the bottom one, you have to see that we fund much less used as genetic models. Not that we are not funding. We do fund actually large animal model, genetic models for human health, but that's much less used actually. So people are not using actually investigators to propose as genetic models for human health or disease. So from genomes, so what do we want now? So we participated in all these genomes of several domestic animal models. So what do we want? So we definitely want the Phaang, that's functional annotation of these genomes, is absolutely necessary for knowing what is conserved and what is not. That's our main goal. So we do need that one. And then we also need genome to phenome correlation. So if you don't have that, we cannot go forward with this program itself. So then the last one, which is the most important for us from NIH perspective, is easy access of data to the investigator. So this is something which is very important. So we all talked about all this data that is available, big data actually. So the important thing is who is using this data? That's the key question. So there are a lot of databases. So who is using this data? And then is it accessible? So how many hits we are getting onto these databases? Basically, that's a crucial question. So we want to know exactly what is happening. So yes, we participated in the genome program, but then, how is it being utilized? Who are the users? So only the people who are, submitting the data into these databases. But then if nobody else is using it, then it's a problem there. So this is something which we want from human genome, sorry. So now, other than that, so how are we participating in programs actually? So are there interagency programs? So right now we have an interagency program through my department. I am part of the program, and Dr. Mark Mirando is here in the audience. So he is the architect of this program. Actually, I wasn't actually. So so under this interagency program, we have research in biomedicine and agriculture using agriculturally important domestic species. So under this program, it should be mutually beneficial. So it should actually impact upon the human medicine or biomedicine, whatever you want to call it. And then it also has to impact upon the animal agriculture or agricultural industry. So that was the main purpose of this program, which started way back in 2009. And then first awards were made in 2010. So these are the priority areas at present. So assisted reproduction and technology, stem cell biology, because that was the hot topic at that point of time, stem cell biology. Then metabolism, and then developmental origin of adult diseases, and infectious diseases and applied immunology. So we have so far funded nearly 30 awards we have made under this program in last 6 years, from both USDA as well as from the National Institutes of Health. So some of the projects of relevance to Phaang objectives, if you look at the first one, control of pre-implantation development, it involves epigenomics, and Pablo is here in the audience. And then we have induced pluripotent stem cells from swine, application to genetic modification. Then we also have systems biology approach to understand endometrial receptivity and pregnancy, and modification of SCNT donor cell metabolism to mimic blastomere metabolism, and finally generation of zoonotic influenza resistant recombinant, there's a mistake there, recombinant pigs via site-directed technology. So these are some of the projects which we have funded. So what's the mechanism available for all the investigators to do any research in the National Institutes of Health is our flagship mechanism, which is known as R01, which probably most of you know about it. So anybody around the world can apply for the R01. There are no nationality restriction or anything. So this is something or you can also collaborate with US investigators and apply for this program. I think I'll stop there. Thank you.
We have a question for Dr. Ravi about the.
Yeah.
Ravi, so the dual purpose for dual benefit is it's in its last year. What's the prognosis for extension? Can you say anything at this point?
Yes. Actually, from the NIH side, NICST is willing to reissue the program. We are waiting for confirmation from USDA, basically. So Mark may want to comment on that.
So I can't give any confirmation that we're going to approve it, but we're working towards that. You know, there is significant interest at USDA NIFA in continuing this program for another 3 years.
Thank you, Mark.
Thank you, and
we hope we are going to reissue.
Good, very good.
And thank you, Dr. Ravi.
Thank you.
Next up is Steve Elsby from the RC UK.
Okay, thanks everyone. I'll be really, really quick because the EU's next, and I love the EU. Okay, I wouldn't want to eat into their time at all, and I mean that honestly. I don't know why everyone's laughing. Okay, so my name's Stephen Elsby. Being really, really quick, I'll talk to anyone who wants to know why I'm here. But I represent Research Councils UK, which is all 7 of the UK research councils, and for the UK and for the US audience, that's effectively NSF plus the basic research parts of DOE and NIH and USDA and various other agencies. Okay, so being quick, BBSRC, who I'm representing here today, love their science. They sent me through about 20 slides like this, going through various different priorities and how they see the genome phenome kind of area. Quite frankly, I like this. This document which came out is very, very good, and everything I've heard from everyone else here, I wouldn't disagree with. In fact, what you've done with this is exactly what we ask our communities to do. So, you know, this is a fantastic start. You've come together, you've started to articulate a common vision, you've started to actually push it forward. Okay, and that's great. If you want to know how engaged we are with this, take the about 20 slides that BBSRC sent me, and then also have a look at the investments that we've made in the past as well. So this is a rich area which BBSRC have been invested in the past, and it's an area where they're looking for more things to come through. The reason why I'm not talking about any specific calls is because we're coming up to a spending review, and as the UK audience will know, that puts abject terror into the hearts of the research councils for a period. We only do it about every 4 or 5 years, so it's an unusual thing for us. We don't do it every year like US people. Okay, so that means that you're not going to see quite so many dedicated solicitations or managed calls being advertised at this moment in time, but this is an area of interest, and the fact that I'm here actually should highlight that this is also an area of interest to BBSRC. There are general mechanisms such as the US Partnership Awards, which exist on our website, which you can have a look at, and I can talk to people more about those later on if they want. So I really only had two things to kind of push through, okay? One of them is probably going to get a big groan from you, but I think Parag actually described it far, far better. You know, I put up basic research challenges clear. Is the impact as well articulated? Actually, Parag's probably got it right. It's that vision. It's that hook, which we as basic research funders find we can engage with this. But if you want to get some of the more mission-led agencies on board, and we would want to see some more of the mission-led agencies on board, you need to give them a hook to actually say, this helps us achieve our mission. Yeah. And so that's really what would be good to work on in this paper, I think. And the other one is quite naturally, as a research funder, we're going to say what can already be done through our existing mechanism. So I think the word backbone has been used here a bit. Okay. What is the core activity which can't be delivered through the individual national funding agencies' existing mechanism? What is the activity that needs to actually be put together to actually link together what's going on already and what individual proposals might do, or what is the backbone activity? And that's the other thing that I was kind of looking for from that document. That's me very, very quickly going through. I could talk about international collaboration. You've seen BBSRC's name up there a lot. You've seen a lot of other international funders up there. You know, we can work together is basically what I would say there. It's just why. Okay. Thank you. That's very quick.
Are there questions for Steve? I had one which is really obvious, but what are the timelines on the crunch that you mentioned from the review?
So the governmental figures come out November 25th, and that will go through to all of our departments. We get funded through one of them, so we're actually an arm's length body. So that's, we actually serve all of the departments in theory across government. So when they get their figure, then they'll then tell us what our figure is around about the March timeframe.
Okay. Other questions? Thank you very much. And the last speaker for this segment will be Jean-Charles Cavit from the European Commission.
Hello, thanks for inviting me to present this speech today. Maybe to start with, as a kind of introduction, I should say, why am I here? If I'm optimistic, I could say maybe because I was together with Steve Capes at the origin of setting up the Animal Biotechnology Working Group within the EU-US Task Force a few years ago. Another argument could be that I was a planning officer and project officer, scientific officer for a number of livestock breeding EU-funded research projects. If I'm less optimistic or pessimistic, I could say probably because you expect me to have some money to put into FANG. And maybe also because I'm the only one you could invite, because I think in the European Commission, in research-wise, I'm probably the only one to work specifically on EU funding for livestock research. So you didn't have much choice to invite somebody else. Anyway, okay, just I will really focus on two aspects. I could have presented what we have done, what we are doing in animal breeding, in genomics of animals. I think this is not that important. But I decided more to focus on two things. One on international cooperation and a possible potential tool that could be relevant to FANG, potentially. And another thing more concrete as a topic on research infrastructure that could also be suitable to FANG. So just not everybody is familiar with the EU system of funding research. One has to remember very easily that EU is not a country. We have a number of member states and associated states, and the funding that comes from the EU framework program is there as a nearly leveraging effect, structuring effect in research throughout European Union and the member states. So what you see as a budget here for Horizon 2020, Horizon 2020 is the framework research program for research and innovation. This money represents probably something like 10% of what is spent throughout different countries in the EU. But as I said, it's a structuring effect. So this program works, started in 2014 and will be run until 2020 with these 79 billion euros for all sectors from aerospace, from to agriculture, health, etc., etc. Basic research to very applied research, etc. In this program, you have basically three big pillars. One is about excellent science, which is where you have the Nobel Prizes research, not necessarily multinational collaborative research. That's where you have infrastructures, one of the two topics I will touch later. You have industrial leadership, and you have societal challenges. The societal challenges is, you have several of them, we will see, and one of them is about agriculture, food, biotech, and aquaculture. And that's where I work. I represent DG Agriculture in a way. I work in a unit dealing with research and innovation in DG Agriculture. A bit less than 2 years ago, I was working in, DG Research, and I worked there for 9 years. Why am I here for DG Agriculture? It's because within this Horizon 2020 program, we are co-managing SC2 that we just see on agriculture and food, etc. We're co-managing with DG Research in the European Commission, certain aspects, and DG Agriculture is more responsible for primary production research, plants, animals, etc. And these societal challenges, you have, I think, 7 of them, we'll see, but one of them is SC2, about agriculture, food, etc., with a budget of 3.8 billion euros, which is around double budget compared to what we could have, what we had in the previous framework program, FP7, which was also a 7 years duration program. So you can see that there is an increase in the budget available for agriculture, food, and biotech. And a significant part of it will go into primary production. The international cooperation, just to say that Horizon 2020, like the previous framework programs, is very open to the world. You have a general opening in the meaning that basically for all topics that are in the work programs, any country can be part of a consortium. It doesn't need to be only EU countries. We still need at least, basically in most cases, 3 EU countries, but then you can have institutes, groups, research groups of any other places. And this is a general opening. And then we have next to this topics where we target particular international cooperation countries and identifying upfront the targeted area and partner country or region. This is the main principle. To give an example, the only one that I will mention as a collaborative project with international cooperation is G+E. This project is very relevant to what FANG is doing. We funded it under the last framework program. In there, you have US partners. And as an exception, these US partners are funded by us, by the Commission, which is exceptional because usually for international cooperation, we fund essentially developing economies partners, not developed economies one, unless there are certain conditions and exceptions where we can do this. But beyond this, I would like to refer, and basically you could have instead of me today 2 other people from the Commission, one to present the International Research Initiative that exists in the health research domain that we have at the EU level, and the other one on research infrastructures. This is managed by DG Research itself. So I have some insight, but I don't have an insider view and heavy expertise on this. But this one, International Research Initiative, are things where the Commission in the health research domain has quite a lot of experience on these. There are several of them we will see. The idea for this international research consortia are to basically identify common strategic goals, agree to share the tasks and the costs, and then you have countries or funding bodies, I should say, funding research according to their funding rules, and there is usually independent evaluation, selection, and funding of projects, but the funded projects work towards common goals, and results, data, and standards are being shared. This is more or less the principle of it. On that slide, you see that each country is working on a specific domain. The name is not there, but you can see it. But in most cases, you have several countries or several funding bodies, institutes working together on a similar domain. That's the cooperation and added value from it. These international health research initiative, you can see there that there are 8 of them. One was mentioned by Paul Fleisig earlier this morning. And the one I will refer to as a model, possibly as an example that could be thought about looking at FANG, is the one on rare diseases, IRDIRC. But you have one maybe that would even be more relevant for FANG by the scope and the focus. That could be, for instance, the International Human Epigenome Consortium. Just to say that the European Commission is usually a member of these existing IRCs. Usually they have been actually, the Commission has been instrumental quite often with the US, NIH, etc., into launching those IRCs. The Commission supports them by EU-funded projects, contributing to deliver on the objectives of these IRCs, and sometimes by a coordination action to support the logistics of these international research consortia. Not in each case, but that happens. Now, if we look very quickly to this International Rare Disease Research Consortium, the idea is cooperation at international level to stimulate, better coordinate, and maximize outputs of rare disease research efforts throughout around the world. The basic principles is to cooperate at international level to stimulate, better coordinate, and maximize output of rare disease research efforts around the world. Teams of public and private organizations investing in rare disease research. There is a commitment, financial commitment from the funders. Research funders with relevant programs have to commit to put at least $10 million over a 5-year period, and then they can join and they become funding members and members of these IRCs. Each organization funds research in its own way, and the funded projects adhere to a common framework. The idea with IRDIRC, for instance, usually those IRCs in the health domain are very focused, a bit like FANG for the moment. With IRDIRC, the idea was 200 therapies and means to diagnose most rare disease by 2020. That's the objective. You have a governance structure in those, even if they are not legal entities as such. You have a governance structure with an executive committee, scientific committees there, and then working groups dealing with certain things. You have there, for instance, ontologies and disease prioritization as a working group in this one, sequencing, model systems, etc. And in the scientific committee, there in IRDIRC, you have 15 members with balanced representation of scientists, patients, industry, etc. The executive committee is usually composed of one representative from each funding body, plus some other, like here you have representatives of umbrella organizations of patients' advocacy groups. So you have the scientific committees agree on a roadmap to reach IRDIRC goals in their scientific area, and the working group recommend actions or solutions to resolve gaps, problems, and difficulties. Here are the committed members, over 40 committed members in IRDIRC, and you see that if you look at the US, you have a lot of institutes directly. It's not necessarily the agencies. You need to have people who can commit money, and it can be the agencies or it can be other structures. You have industry, for instance, Pfizer is mentioned there in North America, partner. You can have charities, etc., etc. But you see that it's quite broad from most continents, Europe, Australia, Asia, with China, Korea, Japan are there, and a lot in the US and Canada as well. And in member states from the EU, you can have the European Commission, but you can have also the member states being represented themselves, like here you Netherlands, France, Italy, UK, Spain. And this is, even if it's not a legal entity, it's quite structured, where you have a governance documents for the structure and the functioning of it, and then the policies and guidelines where you will have all about sharing the data, the standards, etc. And in particular, for IRDIRC, the policies and guidelines, it's a common framework with separate sections for researchers and funders, addressing various aspects of research, such as sharing of data of samples, rapid release of research results, common quality standards, interoperability, involvement of patients. I mean, other than the last bullet, all these above are the kind of things you've been discussing, and FANG is trying to agree on this so far. You have a scientific secretariat for IRDIRC. just to say that in this case, there was a coordination action supported by EU, a contract for 6 years since 2012, support IRDIC to support the logistics of these. Basically, it's to support the meetings of the scientific committee and the working groups. And then what we can say, yes, I mentioned it already. And if you want more information on this research consortium or other ones from the health sector, they are all on the website of the European Commission, probably somewhere else. One thing just to say is FANG looking like an IRC? Main features and success factors for IRCs. Well, it's a soft international coordination of research. It's not a legal entity, but it's based obviously on strong commitment of members with common objectives and requiring collaborative research. If there is no need for collaborative research, then these IRC are not necessarily bringing added value. Common principles and modus operandi and tools as relevant. It's focused on research activities. It's not a network. It's not a talk shop. It's really focused on research activities with measurable outputs. That's one of the key success factors that my colleagues in DG Research Health mentioned, and obviously the sharing of the results. But two things you have to keep in mind about IRCs is you need a governance structure. There is no standard legal requirement for this, but basically all these IRCs have a governance structure with executive committee and scientific committee. And then these IRCs work on the basis of letter of intents from the funding bodies, which commit in this letter to fund during a certain period a minimum threshold amount of money. So I would say, based on this, you need to convince FANG will need to convince the funding bodies, agencies or other ones, about the interest of FANG. But I think you have a number of things there that would be relevant. Now, another one, I will fly over this. It's the second part of my talk. It's about infrastructure. And there is now an open call, basically, or it will soon be open, let's say, sorry, for EU research infrastructures. When we see EU research infrastructures, it's research infrastructures of interest for the EU. It doesn't mean that they have to be 100% based in the EU. They can be partly based in other countries than EU member states. But they are these things to open up key national and regional research infrastructure to all European researchers and to ensure their optimal use and joint development. They will be in the work program 2016-17 that is already published in an indicative way for the moment, but about to be published in the coming days, the official one, where you have this infra code for advanced communities and then another one for starting communities. It would be nice if you could, FANG could have applied to advanced community. The issue is that you cannot apply for this before you had a project under starting community. So for starting communities, these communities have to have never been supported for their research infrastructures integration in the EU FP7 or Horizon 2020. The one thing, you have the budget there, 40 million euros, but this is for a set of projects to be selected. Basically, the EU contribution per proposal will be up to 5 million euros. It's a complete bottom-up approach. So anybody from aerospace to transport to energy to agriculture, etc., can apply. They expect a lot of proposals, and the selection will be quite, the competition will be quite high. I guess something like 1 in 10 might be the final results between proposals and selected projects. It's a two-stage call. One thing important, and we will see that here, is that the deadline for the proposal, the pre-proposal to be submitted, because it is a two-stage approach, is 30th of March 2016. There is not that much time to prepare this. But at the end of the day, the second stage will be only in 17. So the results, whether you are finally successful or not, will be after March 2017. Coming back to this, in integrating activities for these research infrastructures, you have 3 mandatory components you really have to keep in mind: the networking, the transnational virtual access to the infrastructures, and the joint research activities. And basically only 1 proposal per year is expected to be funded. They will be in the domain and not more than 1 project run. the access is to be provided only to key research infrastructures of European interest, able to attract significant numbers of transnational access users, and other national and regional research infrastructures can be involved in other activities of the proposal. I will not go into all these aspects, but if there is an important emphasis put for starting communities on the networking, the standardization, and establishing a common access procedure for transnational or virtual access provision. All those slides are available, so you can look at these. These are logistical details mostly. Now you have here, you see that with 40 million euros up to 5 million euros per project means maybe something like 8 projects could be selected throughout all domains that I mentioned earlier. Now the last, more or less the last slide. First, one thing important is that those international research consortia are not EU funding instruments. They can be created with or without the European Commission. It's just a model of a structure, and as I said earlier, usually the Commission, DG Research, has been instrumental into setting those ones. It's an example. There are 8 of them now in the health domain. I'm now in the process of facilitating the creation of one in the agriculture domain, in animal health. But they can be built without us. But the thing is, of course, they can be created if you have funding bodies willing to contribute. And I would say the objectives have to be a bit commensurate to the level of budget you can expect to get. But they can be created independently. Research topics that we could put in our work programs for collaborative projects, because in a way, once you have the structure like an IRC, you still need projects to contribute to reaching the objectives and fulfilling the gaps. As regards EU for collaborative projects, it could be really wet research, or it could be, like I said, the coordination action to support the logistics. Unfortunately, our work program for 16 and 17 is already basically finalized. And there is nothing specifically for FANG in there. We have drafted it months and months ago. The next opportunities will be to put topics there for the work program 18, 19, and possibly this work program will cover even 3 last years of Horizon 2020. And the design of the topics will start, and our strategy, we are starting to develop our strategy for this work program. And we will really, more concretely, start in 16, probably the whole thing will be completed early in 2017. With a question mark, we don't have the timetable yet, but if it's like we did before, it's like this. And for research infrastructures, you have there, we mentioned already how it was working. And I put potential Horizon 2020 survey. I insist on potential with an asterisk saying there, not a commitment by the EC to support it. I mean, even if I would be happy to support and put topics like this, I'm not the only one to decide. You can imagine we have a lot of inputs from and complicated process to go through. We have to consult with the member states, etc., etc. So don't take it for granted that even if there would be an interesting thing, it would be there. But certainly, the more, in a way, working now in a research unit in DG Agriculture, I think the more you can identify the impact, not only on understanding biology and models for human domain, I think the potential impact that you could figure out on agriculture in general, on livestock production, be it for health, be it for robustness of animals, etc., etc. I think this would certainly help, because we are in a situation where I guess in the US it may be a bit the same situation. We have this excellent research part of Horizon 2020, Blue Sky Research Nobel Prizes, and we have the societal challenges, and they're called societal challenge. Why? Because we want to address these challenges and we want to bring solutions to the market. And FANG is a bit in between. It's not blue sky research, not fully basic research, but we have to figure out how FANG, being successful, it would, in a way, help into improving agriculture and animal agriculture. Now, based on this, IRCs are finished. I'm very, very late, I guess. Based on this, are the IRCs and the infrastructure. You could also think of whether the scope of FANG is appropriate, or could you modulate it either for a proposal under infrastructure, could be bigger, could be whatever, or even for an IRC, the International Research Consortium, it could be fine, strictly speaking, or an adapted solution. Could you link it to, I don't know, phenomics, biomarkers? I don't know, but that's up to you to discuss when you submit the proposals, because of course, for the IRC, it's less obvious, but for infrastructures, probably the impact of what would be done within an infrastructure like this is one important aspect that is taken account when evaluating the proposals. I have to say that I defended, when I was in DG Research before, topics for infrastructures for livestock, which was not so easy to put topics in FP7 for livestock related infrastructures. And we managed to put, for instance, 2 topics on which proposals were submitted, and none was successful. By chance, the plant ones were successful. So in the livestock domain, we were not very successful, or the community was not that successful to put excellent, because there was competition. I wouldn't say the proposals were bad, but there was competition between the areas. Well, the plants and even the aquaculture ones were more successful than the terrestrial livestock. So there are probably lessons to be learned from that. Thank you.
Thank you very much, Jean-Charles.
In the interest of time, we won't take any questions now, but I'm going to get these slides to the both breakout session chairs. So you'll have slides of all the agency speakers. And I'm sure that Jean-Charles will be available for questions in the afternoon. I've arranged to have somebody stay in the room. So we're going to go take the picture now. And the quicker we get the picture done, the quicker you can eat. So you will have nearly a full hour for lunch if we get this done quickly. So Alan is going to stand up and take you to the front steps of the building, which he said he knows where it is. So he's going to take you there, and I'll meet you there in 2 minutes with a camera. So.